Good afternoon, everyone. This is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And if you have any questions, you can start asking. Okay. I think the Rahul is asking. I did not understand the positive and negative selection. So, positive selection means you are actually going to confer the resistance into that particular cell. So, positive means you are going to select that particular cell. Negative means you are actually going to remove all other cells. You are going to remove you're going to remove the that particular cell from the selection. So I think I have taken an example like positive selection means suppose you are actually going to put the antibiotic resistance genes into the uh, the other thing is the Priyanka is asking what type of questions. So at the end of this course, we are going to ask the question of the multiple choice questions or objective type of questions, and yeah, so there you going to have the one question and four options, and it's going to be an objective type questions paper. So Rahul, did you understand the positive and negative selection? Or shall I give you an example, or shall I give you more elaborate explanation also? Okay, I think the Rahul is asking about the explain examples also. Okay, so let me share my screen and then you can be able to understand more nicely. Okay, so so let's first understand the positive selection. Okay, so selection can be positive selection or it could be a negative selection. Now, positive selection means, for example, if I take an example of the ampicillin resistance gene. So, if you have an ampicillin resistance genes in a plasmid, okay, and if you transform that into the host, right? So your host is going to have the ampicillin resistance genes, right? Now, if you take this host and if you plate this host onto a plate where you have added the antibiotics, right? So if you have added the ampicillin, for example, right? So you're going to have when you transform this into the host, you are going to have another cell which is non-transformed. So this is the transformed cells, and this is the untransformed cells, right? And when you plate them, okay, this guy is not going to survive, right? Because this guy does not have the ampicillin resistant genes, whereas this guy is going to survive. So what you are going to get at the end of the selection, right? You're going to get the colonies where this particular clone is going to be present, and that is what it is called as the positive selection. Whereas the negative selection is that for example, if I take an example of that you have, uh, for example, if you take an example of PBR322, okay, so PBR322 has two genes. One is tetracycline gene, the other one is the ampicillin genes. Okay, so if you take the two genes, okay, and if you put the genes in one of the genes, like for example, if I clone the gene into the ampicillin resistance gene, then what will happen is that this is going to be ampicillin sensitive. Okay, now what I'll do is I'll transform this into a plate, right? So I will take it, transform it into a plate, right, which does not contain any antibiotic. Okay, so this is without antibiotic, and I will make a master plate. Okay, so I will make a master plate. So this is a master plate. Then what I'll do is I'll take the master plate and I will plate it onto a tetracycline resistant genes, or tetracycline. I will plate it onto a tetracycline antibiotic, and I will see how many cells are actually going to survive. So the cells which are going to survive are actually going to be those who are going to have the functional tetracycline. Now what I'll do is I'll but so all the ampicillin resistant genes are also going to survive, right? Now what I'll do is I'll take these cells and I will plate it onto the ampicillin. So in that case, what will happen is the some of the gene cells are actually going to be absent, right? Because those are the cells which are actually does not have the ampicillin resistance, right? So now what you're going to see is all these are ampicillin resistance. Okay, so if you go with these kind of analysis, you can be able to say that the cells which are actually going to be ampicillin resistance, see ampicillin sensitive but the tetracycline resistant. So when you put it onto the ampicillin, some of the genes are going to be removed, right? So if you see in the you have two different types of clone, right? One is the only the PBR322, and the other one is the PBR322 containing your gene, right? And your gene is present in the ampicillin. So this means it does not have the ampicillin. So if you see, some of the colonies are going to be disappeared from here, right? Those are actually the colonies which you are looking for, because there in those colonies only you are going to have the clone of your interest. So what you can do is you can just go back to the master plate and then you can look for the colonies which are being disappeared. So this is where you are looking for the colonies which got disappeared. So in the negative selection, you are going to see the colonies which got disappear when you are actually doing the selection pressure. Whereas in the case of positive cloning, it is there the colonies are actually going to appear when you are going to apply the selection pressure. So that is the major difference between the negative versus positive selection. Is it clear now, Rahul? Now Priyanka is asking a hypothetical question. Okay, the hypothetical question is where what she is saying is that can it be possible for us to create a protein artificially using bioinformatic tools in virtual environment? Okay, so and so you cannot create the protein. What you can actually do is you can actually, if you know the amino acid sequence and if there is a suitable template, you can be able to prepare the 3D structure of that particular protein model. Okay, so you can actually be able to prepare the 3D structure of the protein model. And then you can be able to prepare you can do different types of calculations and experiments and all that. And if you want to do that, there are tons of bioinformatic tools or software which are readily available and that you can actually be able to use. Do we have any more questions? Priyanka, is it clear? Or you want to discuss something more? Okay, I think she wants to discuss about the bioinformatics tools, right? So if you want to prepare the 3D structures, one of the easy software that you can use is the modeler, right? Modeler, right now I think it's a ninth version what you can use, or you can use the graphics interface like the easy modeler, right? And that easy modeler is actually going to help you to prepare the 3D structures. Apart from that, you can use the online servers, like you can use the Swiss PDB server, or you can use the so there's so many servers which you can actually use. And after that, you can use the Service server, and that service server is going to have the tools for validating the structure, because once you prepare the 3D structure, you also need to have the validation of the tools, right? So validation of the structure, right? So in the validation, for example, you can actually do the Ramchandran plot, and you can do the verify 3D. So these are some of the plots what you can do. You can do the like errata plot, and you can do the what if and all this. Uh, so these are the things you can do. You can do the pro check, right? So that pro check is going to tell you the accuracy of the structures. Okay, so these are the things you can do, and it's not very difficult. But as I said, these are going to be a virtual, so it's going to be not really it's going to give you the real picture what how the protein is look like, because it depends on the type of template what you are going to use. Is it clear now, Priyanka? Okay, now Rahul is asking another question. If we integrate a recombinant gene in the host chromosomal DNA, is it guaranteed that we will get the protein of our interest? Can anything go wrong later? There are many things which can go wrong. Okay, one of the things which actually can go wrong is the site of integration. So for site of integration is a very important thing or a very important phenomena actually, uh, because if you integrate the gene into a promoter, so site of integration means in what promoter system you are actually going to use for your expression, right? So if you going to use the promoter which is not very good, or site of integration is putting the genes into a into a part of the genome which is not very active, then there will be no expression. So there will be no expression of the gene. For example, if you put the gene into a site of integration into a part of genome which is mostly been remain dormant, or which will or mostly been remain into heterochromatin, then even if you have the gene, it may not be get activated, or it may not be get transcribes, and that's how it may not going to express. The second thing is, suppose the protein got produced, right? So you got the protein, right? That protein may not be stable, right? Because when you are producing the protein was required to have a post-translational modifications, and those post-translational modifications machinery is not available, or you might have done something related to that, and that may make the protein less stable. Okay, so it's going to be less stable, and in that case it's going to cause the protein degradation. So it's complete. So that's what I say. Only cloning a protein and then integrating the protein into the genome is not enough. Okay. For example, some of the proteins, when you put them into the secretory system, they are going to be expressed nicely and they will be going to be recovered nicely. But if you put them into the non-secretory pathway or some other pathway, then it may be problematic for them. Rahul, is it clear now? This session is very limited. Okay, so if you have any questions, you can ask me, and I'll be happy to clear your doubts. I didn't understand the derivatization of the formula in chromatography. Rahul, which formula you are talking about? Divyansh, I think you're right, right? So eight assignments is the best eight assignment what we are going to evaluate to make you to consider you for eligible for the exam. So even if you miss the two assignments, I think it is not a big issue because 8 out of 12 is what we are going to what we are going to consider. So don't worry, you can just continue, keep submitting the assignments, and keep scoring at least 40% marks, so that you will be eligible uh, for the certificate. Rahul, which formula you are talking about? Okay. So if you want to discuss that, so I think you're talking about the resolution of the column, right? Um okay. Let me copy the suitable photo, then I'll tell you. Okay. Uh, do you see the slides? Okay, do you see the slide, right? So what you were asking me is this, right? So if you go to the previous slide, I think that is what we were discussing, right? That you have the different peaks, and A and B is widely separated, so they are actually, this column is good enough to have a resolution so that the A and B get separated, whereas C and D are not been able to separate, so they are actually having the they are not being able to resolve by the column, and that is because the C and D are actually sharing a platform, right? So they are sharing the basis, and that's why they are not being able to separate. And if you want to calculate the resolution of a column, the resolution of a column will be directly proportional to the differences between the peak. So this is what the differences between the peak, and it is going to be inversely proportional to the base of the width. Okay, so average base of the width, which means it's going to be T2 minus T1 divided by W2 plus W1 divided by 2. So narrower to this and bigger to this, the resolution is going to be of a higher side. Now, uh, the resolution is also going to be dependent on to the number of distribution planes, right? So distribution plane can be calculated by the number of the length of the column divided by the height of the distribution plane, right? And this, if you're talking about this formula, okay, this formula, when you are actually going to consider the different types of analytes, okay, so uh, why I have not discussed how you can go from here to here and from here to here is because that requires a large number of the mathematical expressions, and that was not be a part of this particular course. But if you integrate the length, okay, if you consider the length, okay, then you actually can consider that on every distribution plane there could be a molecule which is sitting, and that's why if you want to express this information like L by H, you can also express that information in terms of the T and W, because if you can imagine that if you have a column like this, right, if you have a column like this, right, you can have a distribution planes, right, throughout from the top to bottom, right? And if you consider that one peak is on the distribution plane number one, the other peak is on the distribution plane number two, and that's why if you integrate this information from, you can come back to the this information or this formula, okay? And accordingly you can, if you mathematically process again, then you are going to get the this additional formula. So how we can get these formulas are little more difficult and complicated to understand, but it's all related to, because mathematically you can consider all these planes as the individual peaks which are going to be under ideal conditions you can easily get the uh, molecules which are binding to the each peak, and they can be get eluted. So I think you don't have to worry about how you got these equations, because that needs the complete understanding of how the molecules are going to flow into the column, and how we can actually be able to convert the the length in terms of the retention time versus the width of the peak. Okay, so if you're not comfortable, I think you just remember this, but it's going to take a lot of time if I want to uh, prove that this is this actually. uh, is it clear to you? So this session is very small. Actually, we are going to have the session only for 1 hour. In case you have any more questions, your queries, doubts, you should you should take this opportunity and you should discuss. Okay, because our exam is only next month only, right? So we should, if you have any doubts, you can ask me the questions. Yeah, I think the principle of gene manipulation and genomics, that book is very good actually, for the majority of the part. Like until we were talking about the cloning part, right? And beyond the cloning part, if you want to further study or further want to, read the books, then the chromatographic portions, I think you can easily follow the Wilson Golding or something like that. Okay, and some portion, I think yeah, that is what Priyanka we were talking about. So I think uh, the the initial portion, right? So starting from the module one, okay, the module one to module two actually. So initial one or two modules or three modules actually, you can easily study that part where we were discussing about the cell biology and how the cells are and different types of host and all that, and you can go through any of the standard cell biology book, like you can go with the Lodish or Baltimore or molecular biology of the cell and all those kind of things, right? Then we were talking about the PCR. So for PCR, you can go through the the like the even the Levi's also. And after that, we were talking about the different types of tools and other kinds of things. So and then vectors and selection criteria and all those kind of things. For that, you can easily use this book, The Principle of Gene Manipulation and Genomics. And then chromatography, and subsequent to that, the technical techniques, that how to characterize the product and all that. That for that you can actually be able to use uh, the Wilson and Golding or those kind of biophysics books. And then the last part when we were talking about the applications. So applications, you can easily study from the biotechnology applications and all those kind of books. Overall, I think if you see, there is a course also, NPTEL course, and if you want to get the content, you can study or you can follow that content also. So I have NPTEL lectures also, or like I have the NPTEL. Okay, so let me share my screen and. Okay, so Divyansh is asking about how the restriction enzymes are actually need to be able to recognize the self versus non-self. Okay, so you know that the restriction enzymes are having the three different types of activities. Okay, so you have the S activity, right? Then you have the M activity, and then you have the R activity. So this is, I'm talking about the type 2 restriction enzymes. Okay, so this is the M activity, right? And this is the R activity. Now I'll show you how they are being actually placed. So when you take a DNA, right? So you talk about the DNA like this, right? Uh, so this S actually So at the major at the this major as well as the this grooves area, this S is actually inserting into the DNA. Okay, so when the S is inserting into the DNA, and you know that you have a nucleotides, right? So if I draw the DNA like this, okay, so you have a nucleotide like this, right? And you can imagine this is our uh, coiled, but I'm not showing the coiled, right? So the first thing what will happen is that the S is actually going to enter into the DNA, okay, and it is going to scan, okay, so it's going to scan like whole DNA, okay. If the DNA will not be able to find, if this enzyme is, so you can imagine that you have the M on one M on this side, okay, and the other M is on this side. So you can imagine that the enzyme is going to sit like a clamp, okay. It's like it's like a, like this. You're sitting like a enzyme is going to sit onto the tryptophan enzyme like a clamp, and on this side you are going to have the M activity. Another side, this is also going to have the M activity, and you can imagine that you have the S activity, which is actually going to start reading the nucleotides. If the S could not find, or if S could find a site, okay, for example, if this is the site, okay, so then what will happen? The S will insert, okay, and the M will be in contact to this particular area. So if the M will contact onto this particular area, and both the side, the M is actually going to contact the DNA. If the both side the DNA is, for example, if the DNA is having the methylation, right? So if DNA is methylated, then the methyl group is going to assess that, and because of that, it is going to form the open complex, okay, and it is not going to be get converted into the closed complex, right? uh, Now, if one of their DNA is methylated, the other part of the DNA is not methylated, then this M subunit is actually going to methylate this part of DNA, and that's how this DNA is going to be get protected. Now imagine that if both the sides there is no methylation. So if there will be no methylation, then next to the M, what you have? You have the R, right? Which are sitting on both the sides, okay. So as soon as you don't have the methylation on both the end, then actually it gives the chance to the R subunit to enter into the restriction site, okay. And once the R subunit will enter into the restriction site, it is actually going to form the closed complex, and once it will form the closed complex, the R subunit is actually going to cleave the DNA, and that's how it is actually going to cleave the DNA. Now, the self DNA versus non-self DNA. So self DNA and non-self DNA, okay. So the self DNA is actually going to be either the hemimethylated or the fully methylated. Okay, so that's how the self DNA is actually going to be protected from the action of the recession enzyme, whereas the non-self DNA is going to be non-methylated. So, and once it is non-methylated, it is going to be sensitive for the recession enzyme activity, because they are non-methylated, so they cannot stop the entry of the recession subunits, right? So R subunit into the DNA, and that's how it cannot stop the recession enzyme to not forming the closed complex. Is it clear, Priyank? No, Divyansh. Now, the next question is Rahul is asking if we want to grow the diatoms in culture media, in what form can we provide the silicon? Okay. First of all, I personally don't have any experience of culturing the diatoms, but in my opinion, probably you don't need to provide the silicon into the culture media. Diatoms are actually having the silicon into their cell wall, and that they actually they don't synthesize, but they keep that when they were be in the ocean. So sorry about that, I don't know actually how you can culture the diatoms. So it's only now 5 minutes left for you to Divyansh, you want to discuss something else also, or anything which I have explained but you could not be able to understand? Uh, I think the higher organisms are having the many other kinds of barriers which actually can be much more powerful uh, than the restriction enzyme methods. Okay, what the restriction enzyme is doing is it is not allowing the proliferation of the DNA, right? But in higher organisms, you can easily have the weapons like complements. You can have the defense system like the section, the antibodies. You can also have the defense system in terms of the free radicals. So that's why the higher organisms don't need this kind of system, right? They can easily go with the other kind of, like for example, CRISPR-Cas and all those kind of things are there. But so decision enzymes are very primitive method. Okay. Uh, so it's a very primitive method, okay, that you are working on the sequence of the DNA. But whereas in the higher organisms, you are simply working with the antigens what are present onto the cell wall itself, or cell onto the cells, and that's why you can actually be able to cause a very robust immune response, and you can be able to kill the organisms very efficiently. Divyansh, I think we have already discussed that the dividation part requires very complicated mathematics expression and mathematical expressions, and it also requires the understanding of the many aspects like the how the liquid is flowing, how the the different types of plates are going to be arranged and all that. So I think that is very much beyond the scope of this particular course. In this course, we are only be limited ourselves to the discussion about the recombinant DNA technology and chromatography and all that, rather than discussing about how you derive that particular formula. So we have 1 more minute left before we can wind up this session. If you have any questions, you can ask me. Yes, I think we are going to have the live session 3, which is next month and just before the exam. So you can also ask your queries that time also. So since you all are don't have any more questions, I think I'm thankful for you to that you participated into this live sessions. You have asked your queries, and I'm sure this discussion or the live session could be very helpful for you to clear your doubts. So with this, I would like to wind up my live sessions, and you should keep asking me your queries in the discussion sessions or the discussion board, and I'll be happy to answer. So thank you for your interest in the course of genetic engineering theory and applications.